Assalamu alaikum and welcome to the first workshop of molecular biology and in which we actually talk about the workflow of genetics and molecular biology. So first of all, we talk about the speaker, in which we talk about myself. Aditras is my name, a passionate PS genetics and molecular biology scholar at the University of Health Sciences. Brings a wealth of knowledge and hands-on experience in bioinformatics, stem cell therapy, genomics, genetics and evolution, molecular diagnostics. Then, why genetics? What is the importance of genetics? Actually, it is actually not just a subject. It's not even just a program. It is actually equips the individuals with the knowledge to make informed health and ethical decisions. It's not it's not just a subject or a course. It is actually about the skills development of the individuals. Learning the molecular techniques like PCR, the sequencing, and the cloning provides a valuable technical skill for academic industry careers. Global impact: by understanding and applying the genetic and molecular concept, we can tackle pressing global issues like disease and hunger. Disease and hunger. Now, actually, the genetics and molecular biology is based on central dogma. The major part of genetics and molecular biology is central. It is the backbone of genetics and molecular biology. It is driving the innovations in health, agriculture, and technology. Now we check about the workshop aim, what you gain from the workshop, and it is actually the comprehensive understanding of genetics and molecular biology. Learn the core workflow of genetics and molecular biology from DNA to proteins. Because as we say, this is DNA to proteins, DNA, then RNA, then protein is actually the central dogma. And understanding it is actually we can solve the abundance of problems like cure the diseases and the major environmental factors involved in it. The next thing we can gain from this workshop is hands-on knowledge about the techniques like PCR, sequencing, and their real-world applications. And the next one is skill enhancement. By by have a hands-on knowledge about the PCR sequencing and real-world applications to solve the problems in your area, it actually you can get the skills about it also. Unlock the potential of genetics and molecular biology to shape your life in science. It's actually a life sciences. Okay, now we talk about the workflow of genetics and molecular biology, in which we first of all talk about the sample collection, then we talk about the nucleic acid extraction, in which DNA and RNA is involved. DNA and RNA is involved. Then we quantify the isolated DNA or RNA by using nanodrop and gel electrophoresis. As you know, gel is a old technique, nanodrop is a somewhat latest technique, but if the TN is degraded due to the extraction, in the course of extraction, actually the quantification by the nanodrop gives the result, but the TN is degraded. So we have to shift from the nanodrop to the gel, because at in gel we don't get the quantification of the DNA or RNA, but we can get the quality of the DNA or RNA can be assessed by gel. The next step we can talk about is PCR, in which it is a polymerase chain reaction that is used for the amplification of your targeted gene. We'll talk about it in the course of workshop. Then we'll talk about the sequencing, the most important thing for genotyping, for checking the mutations, and at the end we talk about the data analysis, the getting sequencing. Then at the end we interpret it by using data analysis, in which the bioinformatics has a core role in data analysis. Bioinformatics has a core role in data analysis, in which multiple tools are used, bioinformatic tools are used to analyze the data that we can.
Three types of the micropipettes in the molecular biology labs. We use the P10 or less than P10 micropipettes in which the tips are white. Okay, the tips are white. And the next P100, P200, in which we use the yellow tips, actually greater than P10, greater than P10, we use the yellow tips. And if you use P1000 up to the P1000, then we use blue tips. And for the collection of the buccal uh, DNA or genetic material, we use the buccal swabs, and it is actually the thermocycler. It is actually the thermocycler that is used to amplify the gene of interest. It is used in PCR, and it is centrifuge, and these are reagents used in molecular biology lab, and these are the pipettes. Okay, all right. This is the gel electrophoresis equipments we used in molecular biology lab. That is the power supply, and that is the coaster in which the gel is placed. And this is actually we get after loading the samples, and we get the bands in it. Okay, and the longer bands move slowly. Okay, that is why it is there, and the direction of the movement is from upper to the bottom, but we read it from bottom to upward, because it is actually the we can say it is 5 prime, it is 3 prime in sequencing. And if we see PCR product in it, the small band is here. It shows the small fragment, and it is actually the long band. Okay, now we start from the sample collection. And sample, we can collect the sample from patient's blood. Okay, and also we can collect the sample from ponds in order if we target the microbials or microbiomes, uh, viruses or the bacteria. We can also collect the sample from ponds, and we can also collect the sample from leaves or the soil. Then, what is the purpose of sample collection? It is actually we obtain the biological material containing DNA or RNA. We ultimately need DNA or RNA in order to analyze it, to check the mutations, we check, and the sources of sample collection is human. Maybe it is blood, it is saliva, it is a tissue biopsies, or a hair follicles. By this, we can collect, we can extract the DNA or RNA. In tissue or biopsies, we have to homogenize it because if we don't homogenize it, we can't get the DNA or RNA. So we have to homogenize the tissue. We can also collect the sample from animals, blood, air punches, tissue samples. Also, we collect for DNA or RNA. We can also collect the sample from leaves, seeds, and roots. That is also for DNA or RNA, and also microbes, cultures, and environmental samples. It is actually this one. This one is an environmental samples we collect from the ponds. Okay, now we collect the sample. Okay, now we have to preserve it because if we don't preserve the sample, we degrade the RNA or DNA. Or so if we degrade the RNA or DNA, then we get uh, false negative or the false positive results. That is not actually I need. We have to get the true result. So in order to get the true result, we have to preserve it. Okay. We preserve it in many ways. There are multiple methods to preserve the sample. First of all, we can preserve it by freezing in the liquid nitrogen. Okay. In liquid nitrogen, minus 196 degrees centigrade is temperature. At this, we preserve this sample. Or we using RNA stabilizing agents like RNA letter, also we called it ribolock. It is used to stabilize the RNA. Or we storage in buffer solutions as a TE buffer or a low TE buffer for DNA. By this, DNA can't degrade by DNAs. And we store the sample for short term at 4 degrees centigrade or minus 20 degrees centigrade refrigerators, or we can store for long term at minus 80 degrees centigrade or a liquid nitrogen tanks. Okay. Okay. Now we go to the nucleic acid extraction. After we collect the sample, the next step is nucleic acid extraction. There are multiple methods to extract the nucleic acid, either DNA or RNA. As we say here, the column DNA extraction method, in which the sample we get, that is sample lysis. We talk about the steps of the nucleic acid extraction on the next slides, but it is the general in which the sample we collect, we use the beads. Okay, we can say it beads, or we can say it is filters we use. in which the sample, the nucleic acid or the DNA is bind with the beads or the filter. Okay, then we centrifuge it. The all contaminants or the proteins or the other contaminants like lipids or the proteins are in the bottom and DNA is here. Then we change the apodroph, okay, and then centrifuge it, and the DNA is now into the solution, and it is the purified DNA we extracted. Okay, now we talk about what is the purpose of nucleic acid extraction. Actually, we want pure nucleic acid. We want pure nucleic acid that is free from proteins, lipids, and other contaminants. Okay, there are multiple methods as a manual methods in which DNA can be extracted by phenol chloroform extraction. That is also called a PCI, phenol chloroform and isoamide alcohol. And RNA can be extracted by using trizol reagent. Uh, I will also share the protocol with you of all the manual methods and automated methods in order you can see it and details about that, how to extract DNA or RNA by using manual or automated methods. In automated method, we use silica column based kits as we see here column based. It's a column DNA extraction. It is actually we use the kits in by this we extract the DNA. And the next thing is magnetic bead based purification system in which beads are used to purify or extract the nucleic acid. Okay, now the general steps involved in DNA isolation. The general steps involved in DNA isolation is lysis. First of all, lyse the cell. As we all know, as it is cell. Okay, in the cell is nucleus. In the nucleus is DNA or a chromosome. Chromosome contains DNA. Okay, we have to extract it. If we want to extract it, we have to lyse the cell. When we lyse the cell, the DNA out from cell. Okay, out from cell. For lysis, we use a multiple. We use the multiple chemicals or reagents in order to lyse this cell. In which the first of all is SDS. Okay, it is used to ion balance to maintain the integrity of the DNA sample. Actually, DNA is not degraded at the normal pH of the human body. That is actually the 7. It's more specifically it's a 7.4 to 8.4, it's like that. But at this pH, DNA is not degraded. Okay. But when we lyse the cell, the pH can be changed. Okay. pH could be changed, would be changed, but we have to maintain it by using SDS. Okay. And the next thing is proteinase K. As we all know, the nuclear membrane and cell membrane is made up of protein. Okay, so we break down the nuclear membrane and cell membrane by using proteinase K. Okay, that is actually break down the proteins, even those present in the nucleus and cell membrane. That is present in the nucleus and cell membrane. that can be break down by using proteinase K. So when we lyse the cell, the next thing is the the sample, or we can say the the thing we contain is actually have everything. It is actually have DNA. It is have RNA. It is also have proteins. It is also contain lipids. It is also contain other contaminants. So if we want to extract DNA, so the other things are contaminants. We don't need it. Okay, we don't need it. So in order to remove that contaminants and we purify the DNA, we have to go through this step. Okay, there is a message. We say it first of all. Okay, 9:30. Okay, okay. Now, the removal of contaminants. The contaminants, as we say, that are the contaminants, we have to remove it. We remove it by using PCI, okay, phenol chloroform isoamide. That is actually phase separate the DNA from other contaminants. PCI is a light sensitive, okay. It is it is light sensitive, so we have to wrap it in aluminum foil. And basic function of the I, that is isoamyl alcohol, is to prevent the foaming formation. And the last one step in the nucleic acid extraction is recovery of DNA. The recovery of DNA is done by isopropanol. We uh, we contain now DNA, okay, but it is in the solution form, okay. So we want to precipitate it, okay. The precipitation is done by gel ethanol. Okay, it is used to remove the remaining contaminants present in the DNA. Okay, it is actually RNA. It is not a DNA. It is RNA isolation extraction, in which the collected data, oh sorry, the collected sample is homogenized or lysis. Lysis occur. So, okay, that contains RNA, protein, or a DNA. Okay, then we phase separate it. We remove the contaminants by using the PCI or isoamyl alcohol, by which we separate it. That upper has RNA, okay, and that is contains the DNA or proteins. Okay. Then at the end, we use the isopropanol to precipitate that. By this RNA pellet is formed here, and at the end we resuspend it by using water or a TE buffer, and we collect it for the for further procedures. Now this is the question and answer section, and 5 to 5 minutes break here. And if you anyone have questions about sample collection and nucleic acid extraction, can ask me about that by using chat box or by uh, open your mic to ask the questions. Do you hear me? Anyone have question about the sample collection or nucleic acid extraction can ask me. Okay, the question is why we do DNA extraction. It's really all for. Okay, Mr. or Mrs. Really awful. We extract DNA because it is actually a genetic material. Okay, if if genetic material has mutation or any problem in it, the protein formed by that gene is also affected. Okay, and if the protein is affected by gene that we actually extracted, the problem is we have any disease by this. That is why we extract the DNA. Okay, we will talk about this the next steps as we PCR or the sequencing at which we check the mutations by using DNA. So we have to extract the DNA in order to know about the mutations. Okay, the type of mutations. The next question is the type of mutations. There are multiple types of mutations. There may be a gene mutations or maybe a chromosomal mutations. The chromosomal mutations is that the insertion or the deletions of the part of the chromosome. Okay, if the part of the chromosome is deleted or deleted or inserted, by this the the DNA can be duplicate. Okay, by this the problems can be occur. And if we talk about the gene mutations in which the missense or nonsense mutations or the silent mutations are also occur. In gene mutations, if we talk about the missense mutation, as we all know, I share the screen to all of you in order to answer this. As we all know, DNA contains nucleotides. Okay, DNA contains nucleotides, as we say A, T, G, C, A, G. Okay, that DNA forms amino acids by using genetic codons, gene codons, that using triplets. Okay, triplets. If the one nucleotide is deleted or inserted into it, the there is a shift in the frame. The framework of the amino acids can be shifted, and by this, the protein is formed is premature. Okay, if the protein is premature, we can get disease. Okay, so there is a if DNA or sorry, if the nucleotide is inserted or deleted, the frame shift can be occur, and that is called frame shift mutation. Okay, and if any nucleotide is replaced with another one, and amino acid is changed. So we called it missense mutation. And uh, if one or a two nucleotides are added, and by this the stop codons are produced. Stop codons are actually a UAA in RNA, or we called it TAA in DNA, or UAG or UG, sorry, UGA are the stop codons. And if stop codons occur, the protein will be premature and we can get disease. That is actually called nonsense mutation. Okay, that is actually called nonsense mutations. Also, your answers will be answered. Okay. The next question is, what is the substitute for isopropanol? And the next question is mutation, frame shift, substitution, and subtraction. I don't get it. Subtraction. Can you rewrite it? Really, Opa? Can you rewrite it? Mutation, nonsense. There are same mutations, but okay. The question is, what is the substitute for isopropanol? Yes, if isopropanol is not available, then we can go to the 100% ethanol. We can also use 100% ethanol. By this, precipitation can also be occur. So we can use 100%. If 100% ethanol is not present or is not available, then we can also go to the 95% ethanol. But if isopropanol is not present and we go to the and we use 100% ethanol, there is a difference in the precipitation time. The precipitation time can be changed because isopropanol precipitate in it takes some time. Okay, slightly longer to precipitate by using isopropanol. But if we use 100% ethanol, the precipitation can be occur more quickly. Okay. The next question is, what is the what is the full form of TE buffer? Okay, the full form of TE buffer is tris EDTA, tris EDTA buffer. Okay, that EDTA is actually anticoagulant. Okay, the sample we collect can be coagulated. Okay, that is why we use the EDTA tube, and by this sample is not coagulated. And in by using TE buffer and by using TE buffer, we can also protect the DNA from degradation. Okay, and TE buffer is commonly used in molecular biology to store DNA or RNA. So by this, DNA is not degraded and is provide a stable environment and protects nucleic acid from degradation. So we all know the 1 hour is almost completed. So we have to extend the meeting to next. So I am going to provide you a new link in the chat box. Please connect by it. Okay, I am going to share a new link. Please join it now. Are you here? Please join. Okay. Now, do you hear me? Please tell me in the chat box or by using mic. Do you hear me?
Yes,
Yes, we hear you.
Okay, okay, thank you. Okay, now we're gonna start the next part. I'm gonna share a screen to all of you. Okay, do you see the screen?
yes,
Okay, okay, thank you. So, the next part we're gonna start. Okay, the two parts, as you all know, are completed. Now we have to go to the quantification. The nucleic acid we extract, we use the quantification or the quantity of the DNA or RNA. Okay, and also we have to check the quality of the nucleic acid extraction. The extracted nucleic acid is either a DNA or RNA. We can use two methods. Maybe it's a 10 nanodrop or by using gel. Okay, by using nanodrop, we we have a numerics. We get the result in a numeric data as we think in the one, two, in that way. Okay, and by gel, we just infer the quality of the DNA. Okay, So the limitation of the nanodrop is we can't check the degraded DNA. Okay, if the DNA is degraded, it can also give the quantity of the DNA, but the degraded DNA can't be used to the next part. If we use at the next part, as in the PCR, the PCR result will be zero. Okay, if if the nucleic acid is degraded, the result of the PCR will be zero. So we have to check the degradation of the nucleic acid by using gel, because in the gel the bands will be like a smear will be created, or the contamination, if the contamination is present in the nucleic acid, we can see it by here. It's like a light, that is that is bright light is present in the bands. That is the that is actually the uh, we tells about it, there is a contamination. Okay, so we're gonna start from the quantification. For this quantification, we can do it by using spectrophotometry. Okay, spectrophotometry is a nanodrop. Okay, actually, this is the picture of the nanodrop, in which, and this is the place where we use the where we piped the DNA extracted. The DNA or RNA extracted is piped on that place. So please ensure it to clean that area. Okay, clean that area with the tissue. Okay, if we don't clean that area, uh, the result will be affected. Okay, after this, we get the result there. Okay, the result we get is actually this type of things. Okay, in that place, the result will be this. It's actually a graph. Okay, that graph is actually the salt area, the salt concentration, and that is actually the DNA or RNA concentration, and that is the protein. Protein and salt is actually the contaminations. So it should be low. Okay, the contamination should be low, and DNA and RNA should be high. Okay, protein should be low, protein contamination should be low, and salt should be low, but the DNA or RNA should be high. At that place, the wavelength is 260 and protein is 280. Okay, so 260 and 280 ratio, 280 ratio should be 1.8 to 2.0. Okay, the 260 by 280 ratio should be 1.8 to 2.0. It is the best. If the value of the 260 by 280 is 1.8 to 2.0, the result is best. But 260 by 230, as we say, 260 and 230 should be greater than 2.0. If the value is greater than 2.0, the result is best. Okay, so we start from this 260 by 280 ratio for the purity. Okay, we check the purity. It should be 1.8 to 2.0. It indicates a good DNA purity. Okay. And if the quantity is less than 1.8, there is a protein contamination. And if it is greater than 2.0, it is other contaminations. It may be lipids contamination, it may be salt contamination. Okay, if the quantity is greater than 2.0. And as we see here, 260 by 230, that is actually tells about the salt contamination. It should be greater than 2.0. It indicates no salt contamination. If it is less than 2.0, there is a salt contamination. Okay, there is a salt contamination. Okay, so should be greater than 2.0, and by this there is a no salt contamination. So now we talk about this gel electrophoresis. It is actually the old technique. Okay, then the nanodrop. It is old technique, then nanodrop. First of all, it is. But as we see, the nanodrop has a limitation. Okay, because degraded DNA, degraded DNA can't be uh, quantified by, or the quality can't be checked by nanodrop. Okay, there is a message. We say it first of all. No, there is no question. Okay. So, in order to check the degradation or the quality of the extracted DNA, we use the gel electrophoresis. Okay, a method used to separate the mixed population of DNA and RNA based on size and charge. Okay, as we see, it is the gel and it is the bands. Okay, it is actually the wells in which we pipetted the DNA or RNA. Okay, and by this we get the bands. Okay, by this we get the bands, and it is separated by size and charge. Okay, it moves in that direction, and that is the smallest or the shortest band, and that is the longest band we get. Okay, and DNA migrates through a highly cross-linked agarose matrix in the response to a current. Okay, as we say, we use power supply here. Okay, and it is actually a positive and it is a negative. Sorry, sorry. It is actually a positive and it is actually a negative. So DNA is moved from negative to the positive side. Okay, and it is cross-linked agarose. The gel is actually called a agarose gel. Okay, it is actually a agarose gel. If the percentage, please hear it clearly. If it there is if the percentage of the agarose is high, okay, if the percentage of the agarose is high, the pore size will be small. Okay, the pore size will be small. So it is inversely proportional to each other. Okay, so if we increase the percentage of the agarose, the pore size will be small. So by this small DNA can be go through it easily, but the large DNA is trapped here, and so that is why the large or the longest fragments are here, and the shortest are travel more. Gel is composed of agarose and a GAE buffer. It is also can be a tris phosphate. It can also be a TBE buffer, and it's also can be a TE buffer. And ethidium bromide is used to stain the DNA bands. Ethidium bromide is used to stain the DNA bands. It is also a light sensitive, so we have to use the aluminum foil to protect it. It is actually, as we all know, so we here draw here. DNA has both minor groove and major groove. So ethidium bromide, if we say it is a minor groove, ethidium bromide bind at the minor groove. Okay, and ethidium bromide, when we see in the UV light, by UV light, it produce fluorescence. Okay, it produce fluorescence, and by using fluorescence, we see the bands. Okay, so ethidium bromide is used to see the bands. Okay, now we go at the next. As you all know, agarose gel in the casting tray that is used, that is the casting tray, agarose gel. Okay, and that is the actually the super coiled DNA molecules travel faster as compared to the linear and the nicked molecules. And same size DNA band migrates slower in gel with a higher percentage of agarose. As we all know, we talk about here, if the percentage of the agarose is high, okay, if the percentage of the agarose is high, the same size DNA band migrates slower in the gel. It means the DNA, the large size DNA is moving slowly. Okay, it is moving slowly if the percentage of the agarose is high. It is the actually the apparatus used for agarose gel, as we see in the previous slides also. Okay, the visualization of DNA or RNA. As we also know, ethidium bromide is used to visualize the DNA or RNA. Okay, ethidium bromide is used to visualize the DNA or RNA. But if in the alternative, we can also use the cyber gold. Okay, cyber gold, it is actually a high cost, it's expensive. Ethereum bromide is much, it's not as much expensive as cyber gold or the cyber green. We can also use the cyber green, or we can also use the gel red or a gel green. In gel red, we get the purple type or a red type bands, or we can see in this picture, or we use the gel green, or we also use the cyber green. And why we use that? In order to analyze the gel electrophoresis. Okay, it is analyzed by gel electrophoresis. So It is the supercoiled and linearized molecules, saying in that way. So it supercoiled is not it's our aim. We use this is actually the good plasmid preparation. Okay, it is the good quality we get in the gel. It means there is no degradation and the extracted DNA is good. But if we overload the gel, okay, if we overload the gel, we get that type of bands. It is also not a preferable. And this is the poor preparation. The preparation of the DNA is not good. It is a poor, and we get that type of things. Okay, there is a lot of contamination here. There is a lot of contamination. That is why the brightness of the bands is very high. And as we see here, there is also a contamination. There is also a contamination. See here, and that is also a contamination. That is why there is a smear formation. So next we also use the vertical gel electrophoresis. Okay, in some cases we use denatured. Okay, denatured DNA. As we all know, DNA is double-stranded. Okay, DNA is double-stranded. So we have to denature it into a single strand. Okay, so we use denaturing gradient gel electrophoresis, or also called PAGE, PAGE, PAGE. It is also polyacramide gel electrophoresis. Okay, we use it. It is actually a vertical. Okay, this type of things. It is actually a vertical, and we get that type of results. We get that type of thing. This is actually the apparatus we use. Okay, now anyone has questions about the quantification, nanodrop and gel electrophoresis can ask me in the chat box. There's a 5 minutes to all of you. It's a break time and it is also a question and answers section. So anyone has questions can ask me in the chat box. I think so. None of you have a question. We will start at after 2 minutes. Okay, now we're gonna continue. And I share the screen to all of you. Okay, so as we all know, we talk about the sample collection, nucleic acid extraction, and the quantification. Now we go through the PCR, or also called polymerase chain reaction. Okay, chain reaction, polymerase chain reaction, in which we amplify the DNA. We extracted DNA is amplified by using PCR. It is also called polymerase chain reaction, PCR, and it is actually the thermocycler or the PCR. Okay, the principle of the polymerase chain reaction, a technique used in molecular biology. Okay, it is a technique used in molecular biology and in vitro. What there is a two types in vivo and in vitro. In vivo means in the body. Okay, it means it is in the body, and in vitro means in lab. Okay, in lab, a DNA polymerase, it is actually a more specifically, it's a tag polymerase because why we use tag polymerase because it withstand at the high temperature at 96 degrees centigrade at which we denature the DNA in order to split it into a single-stranded DNA is denatured at 96 degrees centigrade. So we use tag DNA polymerase because it is withstand at 96 degrees centigrade. DNA polymerase used to amplify a piece of DNA by using enzymatic replication. Why it is enzymatic? Because it is used tag polymerase and is used to replicate or the amplify the the DNA. The DNA generated is used as a template for the replication. Okay, as we all know, there is a as we all know there is a two. Okay, there is a two templates or the two fragments. In the next there is a one, two, three, four, and in the next there is a eight. In next there is a 16. In next there is a 32. Okay, there are exponentially increasing in the DNA extracted. The extracted DNA is amplified exponentially. Okay, a chain of reactions start in which the DNA template is exponentially amplified. It amplify even a single piece of DNA to millions or more copies. Okay, the best thing of the polymerase chain reaction is it can amplify even a single piece of DNA to millions or more copies. Okay, we continue the principle. It employs heat-stable DNA polymerase, as we all know, the Taq polymerase. It uses the Taq polymerase, an enzyme isolated from the bacteria Thermus equatis. Okay, it can withstand at actually a 120 degrees centigrade. So at 96 degrees centigrade, at which we denature the DNA, it can also withstand. DNA polymerase enzymatically assembles a new strand DNA. Okay. So as we all know in the previous slide, uh, the template is used to the old strand is actually used as a template to amplify the new strand. That is why it is exponentially increases. Single-stranded DNA used as a template, and DNA oligonucleotides, or we called it primers. Primers are used for initiation of the DNA synthesis. At that point, we get the concept of primers. Actually, the primers are used to initiate the replication. As we all know, the characteristic of the DNA polymerase is the DNA polymerase can't initiate the replication. Okay, as we all know, DNA polymerase can't initiate the replication. So we have to use the primer to initiate the replication. Okay, so we use primers here. It is actually also used to specify the gene of interest we actually want to amplify. As we all know, for example, it is a DNA. Okay, it is a DNA. It is a long DNA, but we want to amplify only that part. Okay, we actually want to amplify just that part of the DNA. So we design the primers at at that point. That is called forward primer. When we design the primers from that forward primer, and at that point, that is a reverse primer. Okay, by this, just that part is amplified. Just that part is amplified. This can't be amplified. Okay, so by this, the gene of interest is amplified, and it uses the thermal cycling. Okay, thermocycler is used in which the heat up the DNA at the 96 degrees centigrade to denature. So that is why it is called thermal cycling. Okay, what what is the requirements? Okay, what are the requirements for the PCR? First of all, we need the DNA template. Okay, the DNA we extracted, the DNA we extracted, we actually need here. Okay, the DNA template is used, and the two type of primers are actually needed here, are required. First of all, the forward primer, and the next part is the reverse primer. Okay, the forward primer and the reverse primer is used in that point. And the next thing is DNA polymerase, that more specifically Taq polymerase is also used at that point, and DNTPs. What does the DNTPs? DNTPs is actually used here in order to replicate the DNA. That is the DNA, okay, and that is actually the primer. Okay, this is the primer, and we need multiple nucleotides, okay, that actually bind here, okay, that actually bind here, that actually bind here. So by this, the DNA is amplified. So we also need DNTPs. So we need DNTPs here, and the buffer solution. Okay, because we have to maintain the pH. We also use. We have to maintain the pH. We also use buffer solutions. And divalent chitons are also used here. And the volume used for PCR is 20 to 150 microliter in small reaction tubes placed in a thermal cycler. Okay, actually, the volume of the reagents, so the whole process is 15 microliter or more than, or can be at 20 microliter, but can also be maximized at the 150 microliter. Okay, and it is used in the PCR tubes. Okay, PCR tubes are actually the smaller than the appendose that are used here in order to placed in the thermocycler. Okay, in order to replicate the DNA. And the thermocycler heats and cools the reaction tubes. Okay, the steps of PCR. When we put them into the thermocycler, okay, when we put them into the thermocycler, the steps occurs in the thermocycler are three steps. First of all, denature, then annealing, and then extraction. First of all, we have to denature the DNA. Okay, we have to denature the DNA into the single strand. Okay, that are double strand, and double strands can be uh, should be denatured into single strand, and the denaturation is occurred at the 94, can be occurred at 96 degrees centigrade. It all depends on you. And annealing. What is annealing? When we get that DNA, now we anneal the primers here, okay, in order to amplify the gene of interest. And annealing is occurred at 54 degrees centigrade, and then extension is occurred. Okay, the new nucleotides or the DNTP is attached here, and the amplifications occur. Okay, and the amplifications occur here at the 72 degrees centigrade and extension. It is actually called extension. Okay, which are repeated 30 to 40 cycles on an automated cycle. Okay, they can heat and cool the reaction mixture in a short time. First of all, it is 94 degrees centigrade, then it goes to the 54 degrees centigrade, and then it goes to the 72 degrees centigrade. So it heat, then it repeats again. Okay, then goes there. So if there is a chain or a cycle occurs. Okay, first of all, it heats up at 94 degrees centigrade, then it cools down, and then again heat. Okay, so these are the steps. First of all, denaturation at 1 minute at 94 degrees centigrade, then annealing 54 degrees centigrade, 45 seconds, and 2 minutes for 72 degrees centigrade for the extensions. And only DNTPs are here. Okay, there are forward and reverse primers are annealed, and at that point, just DNTPs are attached. That are the DNTPs actually attached here to to the extend the DNA. This is actually the GIF in which that that is the extension. Again, denaturation, single strand, double strand is denatured into single strand. Then primer is attached and extension annealing occurs, and then extension occurs. Okay. Now next is the detection of the PCR product. The PCR product we contain, now we have to detect it. Okay. The detection is also done by using gel electrophoresis. Okay. It is also done by using gel electrophoresis to check whether the PCR generated the anticipated DNA fragment. Agarose gel electrophoresis is employed for separation of the PCR products. Okay. The size of the PCR product is determined by comparison with the DNA ladder. Okay. Here, as we all know, this is the gel and this is the wells. Okay. We use here DNA ladder. Okay. This is actually starts from 100, and at that point there is a 500 and 500 base pairs. Actually, this is the 100 base pairs, that are the 500 base pairs, and then it goes to the 1000 base pairs. Okay, that are the known. Okay, that are the known base pairs or the known fragmented DNA. Okay, that is called DNA ladder. It is actually present or available in the um, kit. Okay, it is also called a molecular weight marker, which contains DNA fragments of known size. Okay, this is a known size run on the gel alongside of the PCR products. So, okay, if the PCR product is present here, okay, just beside the 500 base pair, and the next is also present here, and the next is present here, the next is present here. So we know it is present at actually 480, it is actually present at 470, it is present at 600, and it is present at almost at the 200 base pair. By this we infer the PCR product. Okay, okay. First of all, the after mixing all the reagents, we vortex. Okay, we use the vortex. Vortex is used to actually mixing. Okay, the mixing of the reagents. Okay, because the volume of the reagents or the things present in the PCR tubes is very low, so we have to mix it accurately. So we use it by using vortex. After vortex, we placed into the thermocycler. Okay, after 1 to 2 hours later, because it takes almost that time. After it, we piped it the PCR products into the gel. Okay, we placed that into the gel, and after loading into the gel and after giving the power supply and run the gel, we get that type of gel, and we use gel dope. Okay, gel dope. It is actually a UV equipped equipment. By this, we visualize the gel, and that thing we get. Okay, that is that is that thing can be seen here. As we all know, we use the DNA ladder. That is the 400, that is the 200, that is 400, that is the 500. So the PCR product is present just just near to the 500. Okay, so the PCR product is 484. Okay, now we're going to the real-time PCR. It is also called qPCR. Okay, the definition, the real-time monitoring of the amplification reaction. Okay, in the previous one, we can't monitor at the real time. Okay, but at this, as it is a desktop, we can check, it is the graph. Okay, we can check the initial concentration of the DNA. Okay, it is by like that. Okay, it is like that, and it contains the CT value. We talk about it later. The purpose, what is the purpose? To estimate the initial quantity of the specific template DNA. Okay, it is used to estimate the initial quantity of the specific template DNA. By this, we monitor the amplification reaction in the real time. And the advantage of the real time or the qPCR is we don't go through the gel. Okay, no gel electrophoresis can be performed by using qPCR because we monitor the real-time monitoring, so we don't have to go through the gel electrophoresis. By this, we have to use to check the quality or the product size. Okay, so we don't have to go through the gel. Okay, okay. The next thing is principles of the real-time PCR. It is actually sensitive, specific, and reproducible. Okay, the small amount of the PCR product, oh sorry, of the DNA can be amplified by the real-time PCR, and it is very specific. Okay, is the primers we use here is very specific. So the genome trust can be specifically amplified, and it is reproducible. For example, the first of all, sorry, the first get we result. For example, we get the result 400. Okay, 400. And we again perform the same real-time PCR. Again, we also get the same if the condition is here, if the contamination is not present. If the contamination is not here, we can reproduce it. Okay, we reproduce it again, the same result. And the next thing is fluorescence is measured every second. Okay, fluorescence is measured every cycle, and the signal that type of signals is directly proportional to the amount of PCR product. Okay, if that type of things at that point, the amount of DNA is that. Okay, so the signal is directly proportional to the amount of PCR product. Detection of the signals during early phases of reaction. Okay, so the initial amount. Okay, the initial amount of the DNA initial amount of the DNA can be detected by real-time PCR. Curves rises after a number of cycles that is proportional to the initial amount of the DNA template. It means curves are rises, okay, according according to the or the directly proportional to the initial amount of the DNA. Okay, now we go through why real-time PCR. Okay, advantages of PCR. Amplification can be monitored real time. First of all, the main purpose of the real-time PCR or the qPCR is we monitor the amplification at the real time. Okay, we monitor the amplification at real time. Wider dynamic range of up to 1 to 10 fold. Okay, as we all know, the initial amount, for example, is 400. Okay, so we have to uh, if it goes to the 800, it means two-fold change occurs. Okay, and if it goes to the 600, it means three-fold change occurs. And if it goes to the 3200, 3200, it means there is a fold change, four-fold change. Okay, so at that point, it actually measure even actually the 10 raised to the power 10 fold. It can measure at that point. Okay, we can not imagine how many amplification can be occur. So advantages of the real-time PCR is the wide dynamic range of up to 1 to 10 raised to power 10 fold can be amplified by using real-time PCR. And the next thing is results are expressed as numbers. Okay, results are expressed as numbers. We get the amount in numerics. Okay, in that point, we don't get that 484. Okay, we just get the bands. Okay, in previous PCR, in the we get the bands, but in real time we get the results are in numbers. This is also the advantages. Or and the no post PCR processing occurs, so no gel based analysis can be performed in real-time PCR, and it is ultra rapid cycle. Okay, 30 minutes to 200. In just 30 minutes to 200, sorry, 2 hours. In 30 minutes to 2 hours, we get the results. Highly sequence specific and sensitive. In previous slide, I actually we talk about it. It is very specific, sensitive, and it is reproducible. So it is far better than traditional PCR. Okay, real time is far better than traditional PCR. So we actually talk about it again. Requirement of the thousand-fold less RNA than conventional assays. Okay, it means the amount of the RNA or the DNA is very low. I think just a 15 microliter. Okay, so we don't need as much amount. Just a 1 microliter template of the DNA or RNA, and other things are nucleus-free water or the other things. Okay, so we don't need much amount to amplify the DNA or RNA. Confirmation of the specific amplification by melting curve analysis. Okay, uh, in next workshop we actually talk about the data analysis or the analysis of the real-time PCR. Okay, so please also join that workshop also, in which we also actually talk about the analysis of the melt curve analysis, or actually the data analysis of the real-time PCR. We talk about that point because it is very wide type, because as we all need, actually melt curve analysis is occur in real time. Okay, and we examine it or we analyze it by using Microsoft Excel. So it contains a very details. So we talk about it in the next workshop. So it is more specific and sensitive and reproducible, not much more expensive than the conventional PCR. Okay, equipment is costly. Equipment is very costly, but it is not. But the reagents are not as much expensive, so it is not much more expensive than the conventional PCR. Okay, disadvantages of the PCR. If there are a lot of advantages of the real-time PCR, there are also besides the disadvantages of the real-time PCR. Requires expensive equipment. That is also the disadvantages. The expensive equipments. Equipments are very expensive. Okay, due to its extremely high sensitivity, sensitivity is very high. Okay, you may get high deviations of the same experiment. Okay, because contamination is very high. Okay, if there is a little contamination, if there is a little contamination, the result can be affected. Okay, the result can be affected. Thus, the use of internal control genes. So over here we use gap DH. That is actually the housekeeping gene. So we use the housekeeping gene here to amplify it also because it is a positive control. Okay, it always it always amplify. Okay, if if in an experiment is not amplified, it means there is a problem. Okay, if there is a problem, so we don't get the result. So internal control gene is is recommended. Have to use it. Okay, now we're going through the primer design. Okay, it is actually we do it in the third workshop. Okay, we will do it in the third workshop separately. But first of all, we go through it general or in the briefly talk about the primer design. Okay, actually the primers are required. Okay, the primers are required. Why? Because okay, we need gene of interest, and gene of interest can be amplified by using primers. Okay, the primers. So we have to design the primers can be designed by using bioinformatics. Okay, there is a bioinformatics. There is bioinformatics. First of all, mm, there is a identifier or the accession number of your gene. The gene have extension number. So we go through the NCBI Genbank. NCBI is a web page. Okay, we we go here. Okay, and give them the identifier or the extension number so that the coding sequence of the human myoglobin will be amplifying using PCR prediction. The accession number is actually a human myoglobin gene. Okay, so here we get the sequence. Okay, here we get the sequence. Copy that sequence. Copy that sequence. Okay, and paste it into the primer 3. It is separate web page. We open it for using for primers. That is used for primer design. Okay, and paste the copied sequence into it. Okay, paste here and just check. We need pick left primers and left use left primers. Okay, and pick right primers. Because left primers means forward primers and right primers means reverse primers. Okay, select it and then click on pick primers and then click on pick primers. I will briefly talk about it. We talk about in the third workshop briefly. Sorry, describedly. So next, in that point, the targets are that product size will be that, and we can also change there also. Primer size can be changed here. It is actually the optimal size. Okay, the size should be 18 to 20. Why it should be 18 to 20? We'll talk about it later. It is very important to know why is 18 to 20. Briefly, we can say because by this the non-specificity not occur. Okay, non-specificity is very low by using 18 to 20 primer size, and TM should be same. Okay, there is just a 2 to 3 degrees centigrade difference. Okay, between left and right primers. If there is a lot of more than 3 or 5 degrees centigrade difference, uh, the primers not actually amplify the gene of interest. Okay, and the GC content should be That is a minimum or a maximum, but it should be 40 to 60%. Okay, it should be 40 to 60%. Okay, after clicking on the peak primers, we get two primers, left primer and the right primer. Okay, that primer and that. As we say here, the GC content is 40 to 50 degrees centigrade. Oh, sorry, percentage. Okay, 40 to 60%. It is between 40 to 60. And the TM is just 1 degree centigrade difference. Okay, it is also best. Length is 20. It is also 18 to 20. I also say. So it is the best primer. We get best primer. If we don't get best primer, we have to choose in between because in primer 3 we get 1 to 10 combinations of primers. Okay, so we select the best one. Okay, there is a resources primer 3